Sheikh Mohanty, Program Director, SCC Center,
इसी का ना
SCC Cancer Hospital. And Dr. Sheikh is the Program Director and Pan India Head of the Center of the Biology and Biobanking at SCC Cancer Hospital, Bangalore. And he holds a doctorate chemistry from the Indian Institute of Science, Bangalore, and he has completed a postdoctoral fellowship from the
डॉक्टर जो बोल रहे हैं
अच्छा इन पे माइक
Yeah, Dr. Mohanty is also the regional director of the BioBank India Foundation from the South India part, and he is an active member of the BioBank Cancer Care. And Dr. Mohanty is organizing one other conference, in the international conference for the BioBank Cancer Care. I request Dr. Mohanty to come. Please.
Thank you, everyone, and I take this opportunity to welcome all the delegates, all of who have traveled within India and across India. Thanks, Jayesh, for coming across. It's an honor to have you here. And on behalf of BBIF, I also wish that as we scale up the years of BBIF conferences, we think that Indian biobanking diaspora is going to increase with great increments. That's the vision of BBIF, and I'm sure that in developing countries like India, we would take up biobanking as a quintessential critical platform which will reshape disease biomarker discovery. Okay. Now, without wasting time, I would like to go ahead with my talk. So in the next 15 minutes or so, I'll be walking you through this journey as to how multi-omics approaches coupled with recent technological advances have empowered omics to be the front line in tackling the molecular operations in cancer or tumors at the cellular levels, and thereby reshaping the field of personalized medicine. I'm sure at the end of my talk, you also will appreciate that how integrating cancer biobanks or tumor banks with multi-omics can smoothen or facilitate the practice of personalized oncomedicine or precision oncology, as well as help in the easy detection of cancer biomarker and also drug discovery process. Now, before I go into the details of my talk, a brief about the organization I come from. So the name of my organization is Healthcare Global or HCG Cancer Hospitals and Bangalore Institute of Oncology, as it was formerly known as. HCG Cancer Hospitals is the largest chain of cancer care provider in the country, and through its largest network of 26 comprehensive cancer care centers in the country, in India as well as in Africa, it is able to reach millions of cancer patients at their doorstep. Now, we actually follow a hub and spoke model. And the reason I coined this term hub and spoke model is way back in the year 1998, when HCG Cancer Hospital was instituted as Bangalore Institute of Oncology, it was actually initiated as a center of excellence or the hub in Bangalore, with an idea that its spokes or its different other branches of HCG Cancer Hospitals will be spread across geographically to the Tier 2 and Tier 3 cities. And the idea was that when a patient comes to the HCG Cancer Hospitals for the first time for primary diagnosis, and even starting the first time of treatment, eventually the patient would not have to travel across such long kilometers to come to Bangalore. So the idea was that we spread this whole institution to 26 other centers. As you can see here, we have penetrated into 10 different states, and its presence is almost ranges from the eastern part as from Ranchi and Calcutta to all the way towards the south and to the north till Jaipur. So we have a very multicentric approach of HCG Cancer Hospitals, where the idea was, as I said, was to penetrate the Indian geographical cancer diaspora across 26 centers, and then use this opportunity to capture the cancer diversity in the country. So, and also this multicentric approach leads us or helps us to, or gives us a window or an avenue to actually access the wide range of Indian cancer population. As you all know, India is not only the cultural capital of the world, it is also the diverse cancer capital of the world. So, keeping this into account, we also have, I'd just like to mention one thing, that every year we have almost 100,000 new cancer patients who are enrolled in HCG Cancer Hospitals. Yeah. So with this background, I would like to move to the next slide. As you see here, this provides us with a unique cancer patient atlas, where as a patient enters the cancer hospital, as you see on the top, the primary cancer clinical OPD is the place where the patient is enrolled, and these are actually the data touch points where a cancer patient goes to within a cancer hospital or a tertiary cancer hospital like HCG. And we have created a HCG cancer patient atlas to actually capture as much as information from each cancer patient, wherever the patient is undergoing treatment, is undergoing a diagnostic modality, is undergoing a sort of counseling. So each of these data points, wherever the patient goes, is actually a data capture point. And of course, if you see at the bottom, we have, I'll come back to the actual talk when I say what kind of why repository we have established, but to just give you a glimpse of the virtually a journey of a cancer patient in a tertiary cancer hospital, this is actually the patient atlas, which helps us to make a completely structured, a full-fledged cancer database repository. Now, this actually also gives us the handle. Yeah, this also gives us the handle, as you know, that the diverse cancer data also brings in the avenue of diverse omics technologies, diverse opportunities for biomarker discovery, and also because of the complexities of the cancer owing to the advent of oncogenomics and multiomics, I would like to call it. This also gives us the opportunity to not only look at the genome-wide sequencing data, which is already being carried out in the hospital as a routine base for each cancer patient, which drives the opportunity to decide whether a cancer patient is harboring a driver mutation, which will drive the first line of treatment. So, following that, we also are looking forward to discover something what Dr. Sanjay Singh highlighted in the previous talk, is that along with the driver mutations, there is a wide array of newly undiscovered, unexplored, mysterious passenger mutations. Now, these are as you look into the world of cancer vaccines, the era of passenger or co-passenger mutations are those mutations which are lying there, but they are not being explored and are equally contributing to the pathogenesis of cancer. We have actually missed out the bus in actually reporting these co-passenger mutations, which may not be VVS, but they are pathogenic mutations which actually contribute to the phenotype of cancers. Now, that is where the opportunity of window opens for the area of new antigens for developing cancer vaccines. So these are the mutations which are blinded by the doctors, blinded by the genomic scientists, and behind the blind eyes of the clinicians lies these mutations which needs to be discovered. Right. So that is where comes in this thing, and then open the opportunity of tumor microenvironment. You all must be aware of the tumor microenvironment. The cancer cells are surrounded by the microenvironment, which actually determine the response of each of the cancer cell or the tumor towards the drug treatment. Now, along with this, as I said, we are trying to discover each of the possibilities of complexities of cancer, and this, of course, brings us to something which is the Trojan horse of cancer biology, the big cancer database. And that is where we look forward to make a remarkable progress in precision oncology and immuno-oncology, where each cancer patient is actually not only a tumor, as I said, each cancer patient is a lively walking tumor bank and a data repository by itself. So this is where there is generations to come. There is generations of BiobankingConference, generations of multi-conferences, but ultimately we need to develop a atlas which will discover all these mutations. And these mutations are going to be the future cancer-specific targets for vaccines. I'm hoping of a model where breast cancer will not be driven by the Herceptin drug, TNBC, but the cancer vaccine. We are already aware that recently there was the news that ovarian cancer will have its first vaccine in another year's time. Already, I think there are a couple of companies who are looking forward to OvaryVac, which is going to be newly launched ovarian cancer vaccine, the first cancer vaccine which will be launched. So, as I mentioned this diagram, I want to give this prelude. If you see the different omics technologies, the tech developments, they actually have brought us to a situation where we have moved on from a hypothesis-driven, untargeted investigation towards a completely data-driven targeted investigation. And this, what I mean by these words is the focus has changed from collating the data from these
Back
multi-omics
इधर
techniques,
पैनलिस्ट
these
और तो कोई
individual
नहीं आएगा ना,
मैं
मैं
अपना-
personal
हाँ,
cancer
बस
patients' data
इनका भी चलेगा
towards a very focused,
नहीं बस
integrated
थोड़ा देर और
concept
चलेगा
of
उसके बाद
diagnosis,
लंच होगा ना
treatment, and prevention of cancer in individual cancer patients.
लेट हो गया ना उनके
Now, historically, if you look at this, this is a model which was given by Leroy Hood in his P4 medicine model in this paper in 2013. In fact, he actually predicted this that the future of cancer treatment would depend upon triple PM model, the predictive, prevention, and personalized medicine in cancer.
होता
Now, based on
हाथ
this,
से लगा सकते हैं
for example, if
नहीं
you
नहीं ये लाइट
were
ऑन
to
करवाएगा
look
ना
at a particular
ये लाइट ऑन
cancer
होता है
tumor cell
लाइट
and you want to
नॉर्मल
get a complete
उसमें भी
cellular
कभी
readout,
लोग ऑन
what would be
नहीं
the
नहीं वो
approach?
तो ठंडा करना
approach would
पड़ेगा
be to compare take the information from a single cancer
अभी
cell,
होता है तो फिर जाके लाइट ऑन
complete molecular
करवाता
data from
हूँ
a single cancer cell, and compare it with a normal cancer cell, and
ये तो
which
बंद
would be in
पता
the
नहीं
form
कौन
of
कर गया
वहाँ से कंट्रोल हो रहा है ऑन आवाज आ रही है ना लाइट का ही आवाज है ये ठीक है लेकिन कंट्रोल वहाँ से आप कर रहे हैं पहले ऑन नहीं था वो पीछे से ऑन किया
all
जाता
this
है
information is filed together as omics data.
नहीं नहीं
And
पीछे से ऑन
imagine
नहीं
this is
इसलिए
a
आवाज आ
single
रहा है
information
कंट्रोल
from-
पीछे
these
से
are
होता
multi-omics
है ना
information
यहाँ
from
से
a single cell.
कंट्रोल हो रहा है
And
उन्होंने बंद कर दिया
now
होगा
imagine all this is coming up as a plethora of information come from a single tumor and from a single patient, which will represent different cancers from the country and across different species. The Caucasian population patient will be different from an Indian cancer patient. Now this is the height of the multi-omics space. This thing, of course, the challenge in India would be the affordability to develop a multi-omic cancer development because of the challenges of the affordability of a single cancer patient to pay for the different omics technologies. Of course, but there is a future model which are more present in the next slide. But yes, as I said, these multi-omics data are the platforms for developing newer vaccines, new discovery targets, new biomarkers. And this is where these omics data cannot be it is humanly impossible to capture this information and to transform them into biomarkers. So what we need is statistical approaches, artificial intelligence, machine learning to develop patterns, develop new antigens, miss mutations, miss targets, and that is where they all will identify these targets, compare with the existing databases, and come up with newer trends, newer vaccines, newer biomarkers. So this is where humanly it is impossible to collate all this information, but the advantage is when we collate all this information, we're able to not only get the 3 different goals, which are identify novel drug targets based on the newer biomarkers, improve current therapies which are existing, and also discover prognostic, predictive, and diagnostic biomarkers. Now, all these goals ultimately lead to what? You should ask the question: how does this information help a precision oncologist, a genomic scientist? Actually, all this information will help us in stratifying the patient based on the individual patient response. Now, this is where the world of clinical oncology is confused today. Why do patients with same age, mass, triple negative breast cancer respond differently to chemotherapy plus receptive treatment? So, or to a particular drug with the chemotherapy treatment. That is where combination treatments are coming up, and we realize that the combination treatments are coming up with first line of drug treatment, with the second line of drug treatment. Actually the secret to differential response between individual cancer patients lies in these hidden mutations, which are not captured at the first line of genome sequencing, but they're actually the passenger mutations. So, and also we always look at the static information which is lying in the DNA. We don't look at the dynamic information, which where the proteins create havoc in the signaling pathways in the cancer. So whatever we see as static, that information at the genomic level is actually just a static information, but they have to translate into a dynamic information where the signaling pathways are the one where the drugs are targeted by newer drugs are targeted by the pharmaceutical companies. So putting all this into perspective, I thought I'll give this background, but the reason we started this biobank was based on these approaches. And as I said, we have moved on from a traditional one-size-fits-all approach, where all the patients with the same diagnosis, they receive the same treatment. It's like was an umbrella treatment, and historically it was just an umbrella treatment. Okay, patient comes with the same symptoms, but an umbrella treatment was given to all the same patients. But now we have moved to a personalized medicine approach where we are aware, we are conscious, we are alert, we are vigilant, and we are also scared that we would not give the same treatment to the same patient. So this is actually based on the patient's unique genetic profile, where this stratified approach helps us to improve the individual health care of the patient, and most important, the answer is it also brings down the cost of the treatment of the individual cancer patient. So with this, we are moving into an era where the personalized cancer therapy is a term which is now evolving, I would say evolving, and this is a treatment strategy which is based on the idea that, or the ability to predict which cancer patient can respond to specific cancer therapy.
वही
Now,
है कि कोई कॉलेज लेवल पे
the
जो
whole approach of personal
conference
cancer
होता
therapy
है,
is based
event
on,
होता है,
as I said, is
उसका
based on
telecast
founded on
करना
the
है,
approach
करना वो
that tumor biomarkers like DNA,
College fest
RNA,
होता है।
protein, or metabolic profiles are actually associated with patient prognosis and patient
किधर है?
response to drug therapy. In addition to this, the individual patient's genetic
तो
factors
college fest
are now being critically
हर जगह
examined and are becoming
student level पे most
indispensable
तैयारियां होती
for
हैं।
defining how a drug is metabolized in the country, how a drug is metabolized in the patient's body, and how a drug, each tumor cell is responding to the particular drug, as well as how the particular patient is tolerating towards a particular drug. So drug toxicity, drug metabolism, and drug response are actually now dependent upon the unique genetic profile of or the genetic fingerprint of the patient. The earlier term was genetic, now it is genetic and genomic and multi-omic fingerprint, which is defining the drug response, drug tolerance, and drug response. So, based on these background, in fact, this is just a hypothetical futuristic model that I put across that in future, of course, cancer care has been pretty much stagnant, but we'll see a different in next 30 years where, as I said, each cancer patient is a walking tumor bank, and whether it's a tumor, solid tumor, or a body fluid bank, what is the fate of a solid tumor? The solid tumors are sent to the pathology. They are in the form of either a formalin-fixed pathology, paraffin-embedded block, FFP block, or they will be in the form of freshly frozen tumors, or they could be in the form of fresh tumors which are not frozen, but they are freshly available as live tumors. Now, depending upon one of these three situations, whether it's a live tumor, it can be used for making patient-derived xenograft model, it can be used for patient-derived primary cancer cell lines, or it can be used for organoid cultures. Now, routinely, the cancer patients sample is processed, the pathology is FFP block, or they are freshly frozen, or the flash frozen are stored in the liquid nitrogen facility as a cryopreserved specimen. When it's a body fluid, nowadays the era is not the body fluid, whether it's blood, serum, everything we are entering into era where body fluids are no more redundant. There is an era of non-invasive monitoring of the cancer, which is through liquid biopsy. And all this information together, it's a solid tumor or a body fluid bank. What do they need? As I said in the beginning, they need the individual cancer patients' omics approaches where each of the sample, whether you take out the DNA, RNA, or the protein, or do the metabolomics, all the omics approaches are dependent upon what you're extracting from the bispecimen. Whether it's irrespective of whether it's a tumor, solid tumor, or a body fluid, we have now techniques which can differentiate the DNA coming from a solid tumor or the DNA coming from the blood. What I actually mean in genetics is germline versus somatic mutations. So we now need to compare the somatic with the germline mutations, where inhibitors like PARP inhibitors now seem to be working very good in the germline mutations, but when it is a somatic mutation in the tumor, the PARP inhibitors are irrelevant. So that's why you see that PARP inhibitors, olaparib, etc., they're working in ovarian cancer, but they're also working now in the prostate cancer. So we don't know the reason why. The reason is the connection between germline versus somatic mutations. Okay. Now with this background, the idea is that all this needs to be extracted. The omics comes into picture. As I said, multi-omics comes into picture. But as I said, all these needs to be put into AI versus ML platform where we know the trends, where we know that the multi-omics fingerprint from the existing cancer patients can be compared, and we develop what is called as a multi-omics fingerprint of a cancer patient, which will actually define the difference between individual patient response to a particular drug, irrespective of whether patients are age-matched or even the cancer-matched patients. So this is where the next generation looks like. I just put this as a model. The next era of 30 years, we look forward to a multi-omics fingerprint-based cancer therapeutics, where we would hope that, like in the previous one, somebody mentioned that that sequencing has got, the cost of the sequencing has come down. I'm sure the cost of the omics will also come down with increasing the number of patients. Okay. Now this is the flowchart. I'm not going to detail. This is the flowchart of the cancer patients in our repository, and we, as I said, we also collect 3 different kinds of samples. FFP specimen, a frozen specimen which is stored in the liquid nitrogen, and a fresh biopsy which is coming from the OT. All these 3 are stored in the biorepository. If it is a fresh frozen, it is stored in the liquid nitrogen in minus 190 degrees Celsius. And all these are consented samples which are then sent to the EMR, the electronic medical records, and each of the cancer patient data is clinical pathologically annotated and then released for research based on after the review of the HCG Scientific Review Committee and the ethical committee. Okay. On the right hand side, we have also started something which is called as a biobank-driven design of clinical trials. Now, this is not hypothesis-based. The clinical trial, this is a data-based clinical trial which we are starting right now. Based on a retrospective study, if it is a we have like 50 TNBC cancer patients, can the new drug combination given to the same patient, but based on the clinical pathological data which is available, we analyze the data and see, predict that whether the patient data can be used for designing a clinical trial. So it is not a prospective clinical trial, but a retrospective data-based clinical trial, which is based on the retrospective samples which are present in the biorepository. Now, based on this, we have actually, so with all this background, the objective was to set up a patient-centric biorepository. We have 3 different facilities: a cryobank facility, a body fluid bank facility, which is a tumor tissue mass body fluid facility, and a formalin fixed repository. So we have collected now almost 3,000 fresh frozen samples. Plus, we are, in addition to that, we are collecting ultrasonic guided core biopsy, as was mentioned in the previous talk. That core biopsy samples are a very good small sample which can be used for monitoring the patient samples. So, in addition to the core cryobank facility, we have a tumor tissue body fluid bank facility where the same biorepository ID is given to the cryobank sample, which is matched with the body fluid bank sample. So it's a tumor tissue mass body fluid facility. And third is the formalin fixed repository, which is one of the largest in the country. We have now 1.5 lakh patients with 10 lakh patient samples across India, amongst 14 different cities in India, from all the HCG cancer hospitals. And this amounts to the diversity that we're capturing through this biobank. And as you see, this is just to give you that these are the 14 different cities. We have covered only the right-hand side of the India map, geographical map, but the left-hand side has to be captured. But we are at the 50% stage where we captured the FFP blocks from 14 different cities. As you see here, these are the numbers, and I will just not take much time to proceed to the numbers, but we have a center-wide distribution as to which center, geographical center, whether it is Ranchi, Calcutta, or Cuttack, Orissa, has which kind of head and neck cancer patient, what is the number of head and neck cancer patients. In addition to that, we have this. If you see these, the top graph shows the original organ-wise total number of patients across all different cancer sites. The bottom is the number of FFP blocks. In addition to this, what we have done uniquely is we have now gone to a sub-site specific biorepository where you say in the bottommost graph, there are basically 47 different subsites of head and neck cancer based on ICD codes. So the different 47 subsites, that is buccal mucosa, tongue, etc., etc. If you see our data, our buccal mucosa of the head and neck cancer is the largest numbers in India. Then we have the breast cancer samples, where breast cancer samples are histopathology classified as HER2, TNBC, etc., etc. And similarly, we have histopathology classification for not only for this is just an infantry figure, but we have done for almost all the cancer organ specific types. Also, we have the HER2 low expressor, which is a new target for drug pharmaceutical companies. So, with this, I will not take much of time. I'll just wind up in 2 minutes. So, this is a recurrent cancer tumor bank where the recurrent tumor bank is there in the country. All the recurrent cases are also stored. Data associated is also captured. So this is one of the first recurrent tumor bank which is there in the country. And this is one interesting thing. We have a poster for this. Apurva is presenting. We have come across the data that among the top 10 cancer types have diabetes and hypertension together, which are prevalent in at least 35% of the population. So we would like to urge you to go to the poster. This is the impact of coexisting diabetes and hypertension together, and it's insighted from multi-cancer comorbidity data. We have analyzed samples, and we feel that 30% of the population have diabetes and hypertension together in there when we look at the data. Yeah, I'm just winding up. This is the, as I said, in HCG, we actually combine biobanking research, etc., clinical care, tumor board approach, where recurrent cases are discussed. And this is a unique expertise of HCG, which has a pathology, genomics, independent bioinformatics department, a liquid biopsy center, etc. So this all, as I said, combines the whole complete translation cancer bioinformatics discovery division. This is a future plan to develop a tumor cell line biobanking cell, which will be a platform for solid tumor as well as cell line derived, organ derived multi-mix repository. This is a very interesting thing. As I said, HCG, because of its geographical penetrance, we are the future left-hand side shows the good incidence of all the cancer in different states of India. I'll just finish in the last 1 minute. So on the right-hand side shows what we are planning to do a cancer moonshot, where we are capturing the cancer across all the different geographical cities in India. And we are also building something called the cancer medical atlas, not the PCG, based on this. I think I'll wind up in the interest of time. This was just the way. And this is the way we capture the multiomics. Before chemotherapy, after chemotherapy, blood, stool, gut microbiome, everything we will capture and in collaboration with IAC. From the same patient, we are capturing a longitudinal monitoring, multiomics, as well as different sites of blood samples. Yeah, with this, I would like to take your listening. Just a message to all of you that omics is just a technology, biobanking is a platform, but this is the combination, deadly combination, which can bridge the gap between clinical and basic science. And with this, I also want to invite all of you for a conference.
तो कैमरा शूट करना भाई आ जाओ सुबह 7 बजे सनरूफ है ना वो अच्छा भी रहेगा इसके लिए
सॉरी फॉर द लेट will not take any questions.
इसको हम लोग बहुत आगे रख चुके हैं।
We have already started with the data analysis from reading radiomics data, go back to the genomics and biorepository. So all three are combining together to get the radiomics footprint. Thank you, everyone.
माइक ही नहीं चल रहा।
वहाँ पर स्टेज हो रहा है, बंदा क्या है?
So the next speaker is Dr. Dharmendra Kumar. He is senior scientist in ICAR CIRB. For those who don't know what CIRB is, actually the Central Institute for Research on Buffaloes. So he specializes especially in animal biotechnology and holds MPSc and PhD qualification in animal biotechnology with postdoctoral experience. He has had several awards, Best Scientist Award, Young Scientist Award, Best Second Paper
ये चलते
Award, and then
रहने
he has also got recognition in the Limca Book of
चल रहा
Records
बाहर
and India Book of Records.
चलते रहते
He's also associate fellow of the National Academy of Dairy Science.
क्वेश्चनिंग के लिए माइक ही नहीं काम कर रहा, तो क्वेश्चन कैसे करें?
Including me. Dalaji, I think we will have to have one full page of awards, in time to come. So we are eagerly waiting to hear from you.
ऐसा ना हो ना बाद में हमें क्वेश्चन करे
हाँ,
ये अलग हो रहा था, वो
Thank you very much, ma'am. Nice words.
अभी इनके ना बाइट्स भी लेने सबके, ठीक है? फिर में
बाहर ही
बाहर ही ले लेंगे, यहाँ से तो अच्छा बाहर
या यहाँ कोई एक साइड लेके ना
Good afternoon to all. Thank you very much to our
इधर भी कर सकते हैं।
team.
बाइट्स अगर करना है
Thank you, Arab sir, for giving me the opportunity to interact with you and present
भाग
piece
नहीं
of
जाएगा
work. So, my
और जिन जिन का लेना है ना बुला लो, लाइट तो हमारे पास है ही, यहाँ पे लगा के स्टेज के पास यहाँ पे करना है
सेशन नहीं करेंगे,
Madam
सेशन के बीच में कैसे करेंगे?
नहीं, मतलब जब
I'm
करना हो
associated with the
अब मुझे भी नहीं पता कौन कौन है, मुझे नहीं पता वो बैठे हैं।
my model
बस वही
animal
लिस्ट देंगे।
buffalo contributing significant in agriculture GDP. So either in terms of milk production as well as the meat production. So India having a number one in the world in the milk production since 2 decades. And buffalo population
आगे
also
रखने से
less,
ज़्यादा बैठे नहीं
almost 50% of Kerala population they're also contributing 50% in milk production. And India exporting
इन लोग की तो आगे भी
meat to other country of value
इसमें तो
30,000
हम
crore per
शुरुआत यही करते
annually. So with this
यहाँ
much
ये
of
समझ में
production
आया,
of my
यहाँ
model
सब
animal,
जाएगा सब जगह।
I would like to go with the animal somatic cell biobanking.
वहाँ पे आगे आते तो कैसे करते हैं?
So this conference is totally focused on the biobanking. So all we are doing that biobanking, clinical meaning of biobanking is a molecular sample for the classification of preservation of the biological sample is known as the biobanking. So earlier we were focused on the preservation of the germ
close-up पे जाएगा क्या?
cells
चेहरे पे
like sperm and oocyte, which we were considered as the future of the animal or future of the life. But in recent past, so as other biological samples, somatic cells come up as an alternative molecule or alternative biological samples for the cryopreservation point of view.
इन फ्यूचर के
So
लिए
somatic
एक है
cells
जो
is
70-200
biobanking means
और दूसरा
collecting,
एक बाइट
preserving and storing somatic cells,
दो लेंस चाहिए।
mainly either in terms of the skin or muscle or other tissue for the animal for long preservation in the research purpose. So biobanking is knowing that. So what is the advantage of the somatic cell biobanking over the germ cells or other cells? So it's very easy to collect. So collection of the tissue is very easy from the animal. Either you can collect easily from the both of the sex, and also you can obtain from the remote area. So for collecting a small piece of tissue that require around 5 to 6 mm of tissue, and for that it don't require very much big infrastructure. So in very remote area you can collect with a less facility. And apart from that, these cells also you can collect even after the death of the animal. And beauty of that one, the somatic cells are culturable cells. You can multiply them, and it's a dividing nature. So that's not possible with the sperm cell immunocytes. Sperm cells are not culturable, you cannot divide. So what number you have collect, that number will be fixed. Once you have the somatic cells remaining the 10 million on 1 lakh number of cells, you can multiply them
थोड़ा
with
सा
10
और
number of
थोड़ा
cells.
सा
So that's the beauty of
बस
that.
बस बस बस
So
बस
this is the procedure in the right side, mentioned that you can collect the cells from the animal, after that making a
थोड़ा
small
सा
piece of tissue, either mechanically
इनको भी ले ही
or
लीजिए
enzyme
डॉक्टर नंदनी को भी
treatment,
लीजिए
and then
छोटा
put into the culture dish, and after that it will be explanted, it will come out from that
इधर
tissue,
इधर
a small
बस
cells,
एकदम
and
ठीक है
then you can
ठीक
collect
ठीक
for that, and after characterization you can go for the cryopreservation in liquid nitrogen and store for long period, and of course also you can distribute for the
ठीक
researcher and use for the research purpose. So these are the methodology. So why animal somatic cell biobanking is required? So there are a lot of purpose. As Dr. Mohan mentioned about the human cell biobanking. So animal biobanking also equally important, because in India, if you are talking about India having the wild biodiversity of the animals. For example, in a buffalo itself having 21 breed of a buffalo, cattle having 38 breed. So likewise, there's a goat having 40 breed. So likewise, there are multiple diversity of the animal species in India. So we have to conserve them, we have to maintain them for future also. So likewise, if you are having a somatic cell biobanking, then you can use that somatic cell biobanking as a donor cell, and use by using the standard protocol of the SCNT, you can produce an elite animal, you can you produce the blastocysts and use for the research point of view. You can also preserve the endangered species of breed. You can produce animal for the zero transplantation. And also recently gene editing technology has been established and repeatable. So you can edit the cell line, and after that you can use that edited cell line for the production of desired animal or trait-based animal. So these are the uses of the somatic cell biobanking. Apart from that, in recent past also the tissue, ovarian tissue and ovarian tissue preparation also come into the picture, and by that you can also recreate those animals which are having less in number or endangered species. So now I'm just giving some example from the buffalo, what we have did in a couple of years. So I'm just giving, already mentioned that buffalo having 21 registered breed. Out of that, some name are there. So how we are collecting? So you can collect tissue from any part of the body. So normally either you can use collect from the air pinna, which is having less blood flow and it's easy to collect. But difficulty that once you collect from the air pinna, the one scar will be there. So that reduce the aesthetic value of that particular animal. So instead of that, in the recent past, we have standardized that we can collect the tissue from the beneath the tail region, just 5 centimeters above from the anal region. So that there's also having the muscular part, less blood flow, and it is unexposed to the sunlight. So that's giving a more suitability to isolate the somatic cells from these areas. So we have collected different breed of the buffalo, and after collecting, we can go to the standard procedure like already mentioned in the previous slide. So after that, you can go for that. Like we have also checked. So what is their attachment rate, outgrowth rate, when they have attained the confluency, and at what time they attain the confluency after passaging. So in different breed of buffalo, it took around 7 to 12 days, or 7 to 8 days for the giving the outgrowth from the or emerging from the tissue, and after that it takes around 10 to 15 days to attain confluency up to 80 to 90%, and then you can go for the passaging, and then after passaging it take around 2 to 3 days. So this is the time schedule for the emerging of cells, and then you can go for the characterization. So we have used the protein expression characterization like biometin. Biometin is just specific for fibroblast expression, and cytokeratin is a negative marker. So then, or you can also go for the molecular characterization using the PCR approach. So after that, you can go for the multiplication of that cell line, and then you can cryopreserve in a liquid nitrogen for long storage. So after that, we have also checked, intermittently we have checked the post-thaw motility, post-thaw attachment, whether these cells are live or not. They are perfectly maintained to liquid nitrogen. So we check them, their attachment rate after 24 hours, after 4 days. We found that after 24 hours, 60 to 70% cells were viable, and after 4 days, they attain around 90 to 100% confluency. It means
they are perfectly normal and okay. So we have also checked the viability. So we found that their viability is around 70 to 80%. So that's come because the post-thermoplastic also occur after the thawing, freezing thawing, some of the cells are going to die. So if you are attaining 70 to 80%, it means it's normal. So what is the uses of these cryopreserved somatic cells? So we have also tried some of the potential application of these cryopreserved cell line. So as I mentioned that you can use as a donor cell for the SCMT to produce live animal. You can also use the fibroblast cells for the reprogramming to induce pluripotent stem cells. Later on, you can differentiate into any cell lineage. Also used for the gene editing purpose, and also you can use for the basic research, and it's also a raw material for the isolation of DNA, RNA, protein, because it's a live cells and dividing cells. So I'm just giving some example of these all. So we have utilized the cell line from the our repository from the two different breeds, and we found that for the SCMT process, and we found that they have the cleavage rate, the blastocyst production rate is within the range. We have also transferred into the animal, and recently we have produced 10 clone calf out of these fibroblast cells. Also, we have utilized the cells for the gene editing. So we are utilizing the gene editing for the myostatin. So myostatin is a gene which is a negatively regulating the skeletal muscle mass. So if you are mutating that myostatin gene, so their muscle mass will be increased, so the milk production will be increased. So lesser period of time, the animal will attain the more growth. In that way, you can increase the milk production. So with this purpose, we have also edited. So currently, we are having the 5 edited cell line in our repository, and we are utilizing that edited cell for the SCMT, and we have transferred some of the embryo blastocysts into the animal, and we could establish 2 pregnancies. But unfortunately, all these 2 pregnancies have been terminated at the during the month of 6 months of the pregnancy. But we are transferring some animal, their pregnancy is due. So apart from that, we have utilized that cells for the reprogramming into the induced pluripotent stem cells. So we are after introducing the transposon-mediated 6 reprogramming factors. So we have utilized after introducing that 6 reprogramming factors into the fibroblast cells, and after 20 to 25 days, that fibroblast cells converted to the stem cell-like property, and they have shown all the character of the stem cell-like property, and they have differentiated into different lineages. So that paper has been also published. And also we have, so we all know that, so cryopreservation and thawing process is the process where it's reducing the viability and generating a lot of free radicals in oxidation process. So for the reducing the oxidation process and increase the viability, we have used unique antioxidant molecule, that's microtube. Normal antioxidant is not able to penetrate into the wall of the mitochondria, and mitochondria is the powerhouse cells where all the ATP formation takes place. So during that process, there are a lot of free radicals have been generated. So normal antioxidant not penetrated to the matrix of the mitochondria, and they are not work that efficient. So mitochondria have a 100 times more potency, and they are reach to the site of action, and they reduce the ROS production. So at the concentration of 0.1 to 0.5 micromolar for the 72-hour treatment, increase the viability by reducing the ROS production. So you can also use that cell line for the research purpose and basic research. So one minute. So apart from that, that was the buffalo story. So far in India, NVGR, so National Genetics Animal National Bureau of Animal Genetics is situated at Karnal, is a nodal agency. They are maintaining the cryopreservation, or they are maintaining the biodiversity of other species. So far, of 9 domestic animals, 32 breeds are having their cryopreserved the somatic cells. And out of that, 8 we have contributed from the buffalo. So apart from that, so wildlife also is an important segment of the animal science. So in a wildlife, Lycon is a subsidiary of our organization of CCMB in Hyderabad. So they are nodal agency for the cryopreservation of the wildlife germ plant. So apart from that, other zoo like the Wildlife Institute of India, they are focusing the Bengal tiger, Asiatic lion. Similarly, Alipur Zoo Garden, Kolkata, Mysore, and other agencies are also involved into the maintaining the biodiversity of the animals, wildlife animals. So far, Lycon has cryopreserved 27 fibroblast cell lines from the different wildlife, and they have utilized the semen of the crapula wildlife for the AI, and they have produced one live animal out of that. So apart from that, this biobanking also has some certain challenges. So challenges like technical challenges, funding and infrastructure, regulatory and ethical issues, and limited awareness and expertise. Because the animal science having very limited facility and resources are there. If we are talking about the cloning, so only 2 laboratories working for the cloning for the entire nation. So we need a lot of investment, awareness, and all the things. And apart from that, definitely these are also having a lot of perspective for the integration of this technology, the cloning and genetic engineering. Then also the public-private partnership is required expansion of wildlife conservation to field. Agencies are only involved. So these are some challenges and perspective. So this much I'm given to them. Thank you very much. I hope I try to maintain my time.
We won't be able to take questions right now. Thank you.
Yeah, thank you.
So our next speaker is Dr. Nirala Ramchiri. He is a professor at the School of Life Sciences, Jawaharlal Nehru University. He holds a PhD from Department of Genetics, Delhi University South Campus, and Master from University of Agricultural Science, Harvard. His research focuses on comparative functional and evolutionary genomics of capsulosis, particularly in the area of biosynthesis. Professor Ramchari has authored novel publication, and he has given many invited lectures, and he has many awards. So, Dr. Nirala.
So while this is being tested, if you have one question for the previous speaker.
Good afternoon, everyone, and thank you. Thank you so much. At the outset, I really thank the organizer, especially Vita, for inviting me to share some of the work that we have been doing for the last couple of years. So, I'll be talking about plant genetic resources, mainly brassica and capsicum. Even though I have written less broad topic, there's a plant genetic resources. The molecular tools that have been used is about molecular markers like various DNA markers. Then we do use the the metabolites as well as genomics derive some of the DNA markers that have been used for the identification of core collection of or unique collection of the genetic material in plants, especially brassica and capsicum. So this is the Korea Brassica Genome Resource Bank where I worked for for almost 4 years. Professor Yeong-Pyo Lee, Department of Horticulture, Chungnam National University, where he had large collection of Brassica germplasm, including the genomic resources like Bag Library. Earlier, before the advent of the next generation sequencing machine, there was people were using that Bag clone for sequencing the genome of Brassica. So if you look at Brassica, there are so there are several species under Brassicaceae genus family, and among these more than 35 Brassica species are known, of which our cultivated crops like cabbage, cauliflower, Brassica napus, and Brassica in India, and Brassica oleracea, they are known for this cultivated as a crop plants. So if you look at here, the cultivated Brassica, there are 6 species. One is tria diploid, and tria polyploid derived from the diploid species, and they are derived from the originated from the same family with the Arabidopsis. So they are the mustard oil that we consume every day is MP diploid. This is the A and B genome, and derived from this one. And if you look at the Brassica rapa species that are being cultivated in China, Korea, and Japan for development of kimchi and other pickles material in those countries. So Korea Brassica Genome Resource Bank started with 1993 construction of secreted mapping population. At that time, the mapping DNA marker was recently developed. Then Professor Yong Lim developed that mapping population to develop genetic map, because that time there was no genetic map, and then eventually that genetic map was used for the construction of high density genetic map for the whole genome sequencing. And in 2002 and 2003, the genomic based genomic resources were developed, and I joined in his lab in 2008, and part of my work was to develop a bag and sequence DNA markers, so that the markers that we develop can be used for the alignment assembly of the genome sequences of Brassica rapa. So the Korea Brassica Genome Resource Bank contains till now that 2,77,440 bag library, cDNA clones 91,000. Seed banks is the most important thing that are collected from different countries, especially in Asian countries like China, Korea, and Japan, where they have collected around more than 3,000 Chinese cabbage, the heading cabbage, as well as the other Brassica oleracea cabbage, cauliflower, and Brassica napus and Brassica oleracea, and developed around 1,400 molecular markers that were being used later, and those collected germplasm to genotype whether they are duplicated or not duplicated. So that not only collected the germplasm seed bank, because unlike the animals, so we can collect the seeds and deposit in the seed bank and then grow and evaluate. Unless we evaluate it phenotypically as well as genetically, then there is no use of this. So in Korea, it is very very cold climate. That's why they always grow plants in the polyhouse of the greenhouses. Is the evaluation, phenotypic evaluation. One is that Brassicas are highly heterozygous. So that's why to produce the homozygous lines to be sequenced or to get more genetically derived plants, we need to have spore microscope culture, which is haploid. Otherwise, if you culture through processing treatment, the immediate next generation will get homozygous lines. So this is the multiplication of seeds and field evaluation of the different traits in Brassica, Chinese cabbage as well as Brassica napus, and I was there, and then we did lots of analysis of phenotypically different Brassica species with respect to nutrient content, with respect to DNA marker, using around 1,000 different collected Brassica rapa species evaluated by me, and those phenotypically characterized the traits or genotypes that are documented. And once the seeds are collected, we store here like this in a packet for 5 to 10 years. Then we regrow it and multiply it, distribute to them. And this is the one where the bag, derived library is deposited in ultra deep freezer. So the collection and trait evaluation, chemical component analysis and selection of lines, microscope culture. These are the seed banks or genomic resource banks' main idea of all the collected around 6,000 accessions that are from different species. So not only that, when 1,000 different genotypes were evaluated, we evaluated with different nutrient component and different phenotypic traits or different components of important mineral elements of vitamins like this one. Since I already told you that brassica, Rapa are very highly heterozygous, so we have done double haploid culture so that it can be maintained, in homozygous condition to utilize in the next breeding program. So this is the lutein content we analyzed, and this is only a few samples that I have shown in the slide. Glucobrassicin, it's a kind of glucosinolate which helps in anti-cancer or cancer prevention. So we did analyze the phenotypic trait also, different phenotypic in brassica also we have characterized, and this is the different Chinese caves. So you see in the not exactly caves, this is widely grown in Korea, China, and Japan. So first assignment that I got was to develop markers and genome-wide markers. There are 12 chromosomes in brassica. Then out of 12 chromosomes, we have taken the uniformly distributed molecular markers for every chromosome, so that you can analyze in the 1,000 brassica, Raba Chinese caves germplasm. We developed, and we first developed the EST-derived molecular markers from the coding region. The SSR marker is nothing but a simple sequence repeat where you get lots of repetition of the DNA sequences, nucleotide sequences in the genome is most prevalent in case of plants. So we developed around 1,500 more robust markers. Then we did classify the brassica based on the molecular markers that are distributed in 12 chromosomes or the whole genome, and we took 36 different capsicum germplasm. That are wild type also there because it's been known by screening that the wild type germplasm are very useful for the breeding of resistant trait, but problem is that they are not crossable. Just initially we try to understand whether the different diseases are not affected. Those lines are not affected by the different diseases, and we did analyze. This is the different brassica, wild type brassica, plants that we got, and it is deposited in a seed bank. And those markers developed from the brassica rapa was also used in the different wild type species. We sequenced the marker region in the SSR region. We found a highly conservation of the markers and in different wild species, saying that it is highly conserved. And we did phylogenetic analysis found it closely with this species. So again, we resequenced because by the time SIPO, one genotype was sequenced. And while screening the germplasm, 1,000 germplasm, we found that some are disease resistant, downy mildew, some are clapper resistant. We resequenced 10 different brassica rapa lines. We did develop SNP markers based on genes. We developed insertion deletion markers so that those markers can be used for mapping those traits and eventual identification of genes for the trait that are resistant to different traits. So finally, we developed more than 1 million SNP markers and more than 1.2 million indel markers in different genotypes, and those markers are being used for the genotyping of large number of germplasm. And we identified around 250 core collection non-duplicated different genotypes from 1,000 different Chinese lines. So again, when I was about to come,
मैं देखा था लिखा था कि चार्ज भी करता है
lines were resequenced by dealing
फोर के नहीं
Korea, and did lots of presences because it's more widely used markers for the identification of So by doing this, we also tried for identification of genes or markers that are related to the showing the resistant to different diseases. One disease that is a clapper disease. We map from the resequencing data as well as the molecular marker data. From two individuals, we map this one from one gene and this is characterized. And apart from that, brassica rapa, we find a diamond resistant genes in germplasm. This is insect. Then we map and develop molecular markers where the susceptible resistant, there are distinct distribution of molecular markers that are highly linked to the trait of interest. It has been published. Apart from that, we did analyze the head splitting, means when the KBS head splits, then commercially it is not viable. So we develop molecular markers for heading resistant trait, excluding resistant trait. This is what we developed from our germplasm. Apart from that, we also from the germplasm radius collection, we did analyze the fusion with resistance and develop markers and map for the trait of interest. So now coming to the current research that I have been doing, that we are working on the capsicum genetic resources. Like we collect, capsicum from all over India, and especially I'm working on the ghost chili, known for very world famous in 2007. It was known as the world's highest natural occurring capsicum species. So we look at the origin of the capsicum. There are different groups: capsicum annum complex, capsicum annum process, and capsicum chinese, the fusion complex, and then bacadum complex. So if you look at that, this out of this around 35 capsicum species known to exist, only 5 species are cultivated, and out of 5 we are working on the 3 capsicum species. And this is the Better India map. If you look at any of the information you want to have, Better India has a very good idea of displaying where you get the chili map. And this is the ghost chili or Bhujalokia. We say that this is the highest pungency containing chili, and we have from northeastern India also several chili, and we have throughout India different traditional cultivars of chilies are there. So we have collected all that, and those chilies from northeast India, 2012, it is known as capsicum frutescens. There is small, upright fruiting capsicum. It, Mizoram got GI tag in 2012 itself, and ghost chili, which is also known as Moris in Nagaland state of India, northeastern states, they got in 2017 the GI tag. And this Dali Kursani, Sikkim also, they have very spicy capsicum chili that we have collected, and also they got in 2020 the GI tag. So my lab is concentrated on the ghost because the ghost, it is known as the liger, earlier, boat means bigger. Bigger means now become the, boot. Boot means it is from bigger to the ghost. So they have depicted in different, cartoons. Like one movie is English movie is a Ghost Rider. Whenever you ride a bike, all the, road burns. So it is, if you look at the ghost chili and Bhujalokia and the internet, you will find a different, cartoons. Being widely used. And where does it stand in terms of spiciness? The pungency, the measurement of pungency was developed by, technique was developed by Scoville. So we measure the spiciness of the chili in, Scoville unit. So our, ghost chili lies here, 1.5, million Scoville unit compared to others. Using this, our material in USA, Mexico State University, they have developed more higher pungency before we do this one. So when I was South Korea, it became very world famous. I was from the northeast India. I tried to work on this one road in that. So what is the use of traditional folk medicine, especially in fever and scaring away the elephant? And so many world famous for Guinness based on the ghost chili. This Gordon Ramsay from famous UK cook came. This lady created world record. And this from 2020, the left corner, this is not visible. He created world record by consuming from Canada. The right of, right bottom. He tried to create a world record, but his esophagus burned out. He has to hospitalize by eating the ghost chili. So now 2009 itself, Defense Research Development Organization, the India adopted this chili as the, the material to develop a chili grenades to catch up to be adopted in the defense system. So the first successful use of this ghost chili grenade was against this fellow, Sajid Ahmad. In 2015, he came across from Pakistan, and then he was fighting a competition or something with the Indian Army. And Indian Army thought they were chili grenade was right. After some time, they didn't get any noise, so Indian Army went and got it. If I cried chili, I was sleeping up here. My students have lots of problem for doing research on this. So apart from that, there are lots of use. Before you knew, if I know 2012, I went to Canada and the Dublin side. They grew this, they grew the plants and then they have to sell it there. So lots of use of this, use of this ghost chili, including vodka and commercial product. But being a Northeastern or Indian, we didn't know how to use this program. So this is the most use this one. So our now current is that we collected around 286 samples in genetic bank. We did evaluate phenotype for GCMS, LCMS. We did analyze the phenotypic trait also. We did material content also, collected lots of seed bank we have developed. We grow and multiply the seeds. And currently we're working on the development of firstly genomic resources, molecular markers, epigenomic markers. We have proteomic done, we have metabolomics done, we have done on the trait capsaicinoids, which is known for causing pungency, carotenoids, vitamins, and mineral elements. So we did, firstly because there was no genomic resources, we developed molecular markers around, 5,000 molecular markers from each species that was used to, genotype, and we classify different capsicum species based on these markers, and redundant genotype have been used. Now we have 101, non-redundant, genotype of Gosili, another capsicum species. We develop non-coding markers also. Then lots of markers are being developed, and then same again the non-redundant, genotype were, found to be exactly associated. Then we found finally this is a capsaicinoid pungency spinase, just 1 minute. So we have, developed molecular markers. We did analyze GCMS metabolic content. We found that not only higher content of, spiciness, but there are genotype which shows the lower content of, spiciness. So we developed molecular marker for the transcriptome sequencing also. We did analyze the expression of some of the genes, because all the biosensitive pathway of genes that are known to the capsicum, known as the common CILD, is very highly expressed into that. There's two highly expressed, the highly pungent capsicum species. That's why we suggest that because of this highly expression of around 10 genes, very high overexpression, we get extreme spiciness. We develop molecular markers based on this one only. There's a high and low pungency. Now these markers are being used and validated for breeding the high pungency in any of the capsicum species. We did analyze the mineral element content also. Now we have sequenced the whole genome, as well as 10, different genome of 5 each from capsicum annum, capsicum fructescens, and capsicum glossili. Resequencing also, we have developed molecular markers, indel in SLTs mostly. We are going for an indel only, and this is what I wanted to present. So, a lot of brassica work that I did when I was in South Korea, Professor Sumna, Professor Yongkyo Lim, Sumna National University, and Indian funding was provided by Department of Biotechnology, University Grants Commission, and Science and Engineering Research Board, which is registered as the Honors Research National Research Foundation. And these are the students who did and went somewhere to, work further. Thank you so much.
Thank you very much. Lack of time, you can ask him questions during the
हाँ? नहीं, बोलने दो।
So now I would request our chairpersons to kindly felicitate our speakers of this session, Dr. Mohanty, Dr. Dharmendra, and Professor Nirala. Dr. Mohanty, please come on back.
हाथ दिखा देना, हाथ दिखा देना, हाथ दिखा
फोटो फोन
बोलिए।
वीडियो हो रहा है, ऊपर तो वही।
हाँ, क्लोज हो रहा है ना, तो आप इधर ही देखेंगे। हाँ दिखा दो।
Professor Nirala, please. Thank you, Nandini ma'am and Dr. Yadav. So with this, we'll have quickly half an hour lunch break. and will join for the next session. So kindly bear with us because of the time constraints, things are moving a little faster. Thank you.
चल भाई खा ले बंद कर कूपन लेना पड़ेगा ना कूपन लेना पड़ेगा